Hello everyone. We are glad to continue our seminar series, Applications of Engineering in spring semester. And we have a very good program this semester. We have speakers from different disciplines, and they will talk about their scientific work in applications of engineering. And today our first speaker is Mete Civelek, and we would like to thank him for accepting our invitation. So Mete Hocam, Başak Hocam, you can go on if you like.
Well, yes, thank you. So I've known Mete from a long time ago, since we were undergraduate students in Boğaziçi University in chemical engineering department, and Mete was one of the smartest kids during those years. So I'm very excited to host him today and hear about his work. After graduating from Boğaziçi University, Mete moved to United States, to Pennsylvania, to get his master's degree and later a PhD from University of Pennsylvania. And then he moved to California for a postdoctoral fellowship. And then currently he's in University of Virginia, and he's an associate professor in biomedical engineering, and also a resident faculty for public health genomics at the university. He's the chair of Diversity, Equity, and Inclusion Committee at the department, and also a fellow of the Genomic and Precision Medicine Council of the American Heart Association. So without taking much time, I will leave the floor to Mete, and join me in welcoming him. Thank you, Mete. It's all yours.
Çok teşekkür ederim. It's a really great honor to be here. Yes, I've known Başak for a long time. We're no longer kids, but it's still wonderful to see all of you. Thank you so much for coming. And also for those of you who will be watching this later on, maybe on recording on YouTube, if you have any questions, feel free to email me with your questions. I'll be happy to engage with you. So I'm going to talk to you today about my lab's work on using this approach called systems genetics to understand vascular smooth muscle cell function. The reason we care about this is because the cardiovascular disease is actually the #1 killer in the world. This is based on statistics from the World Health Organization. And cardiovascular disease encompasses the coronary artery disease and heart failure, but coronary artery disease specifically make up the largest proportion of these deaths. And what it is essentially the blockage of the main artery that provides, blood to your heart muscle so that it can keep pumping heart. And in advanced stages of the disease, this can lead to significant narrowing of the blood vessels. As you will see in the next slide, the underlying pathology is a process that we call atherosclerosis. So let's first focus on this top part. This is a healthy artery. It contains 3 different layers. The innermost layer is a single layer of cells called endothelial cells. And these cells are very important because they provide an anticoagulant surface so the blood can flow without clogging, right, the arteries. The second layer is a layer called media, which consists of smooth muscle cells. And these cells are also really important because they basically contract and relax and adjust your blood pressure. And the outer layer is another layer called adventitia, and this layer provides structural integrity to the blood vessels, to the arteries. So even in very early ages, starting at the age of 5 and 6 and 7, these lipid deposits start to accumulate in this layer right below the endothelial layer. And this is really cholesterol, basically, that's accumulating in these layers. And as we all age, this becomes larger and larger and creates this atherosclerotic plaque, you can see clearly that there are primarily two types of atherosclerotic plaque. One has this really rich lipid core, and the other one has a smaller lipid core and a thicker layer of these smooth muscle cells. Now, those of you who are mechanical engineers are probably immediately thinking about the stability, right? And this has a lot of structural integrity, this thick layer, whereas this thin layer is really vulnerable to rupture. And when this rupture occurs, you have this blood coagulation that really clogs the artery and then limits the blood flow to the cardiomyocytes, to the muscle cells in your heart. And this is basically a heart attack that occurs. If this happens in your cerebral vasculature, in your brain, then this leads to stroke, basically. But the underlying cause for both of these diseases is atherosclerosis. And I'm going to focus with a cross-section of an artery to this phenomena that we call the stable and the vulnerable lesion, because we all have actually lesions, whether we like to admit it or not, in our arteries now. But we hope that they are stable lesions and they are not these vulnerable lesions. And what determines the lesion integrity is basically the number of smooth muscle cells that have migrated from the media to create this thick fibrous cap. And they have also secreted these proteins that we call extracellular matrix proteins to create essentially a thick, structurally integral atherosclerotic lesion. Whereas in this case, as you can see, this is a thin lesion which is prone to rupture. So what are the risk factors for this coronary artery disease and the underlying cause atherosclerosis? As most of you can, I'm sure, figure out, is there's both an environmental and a genetic component. So the environmental component are things like your lifestyle, your smoking, obesity, high blood pressure, etc. But there's also a strong genetic component. And that's actually why most of the time when you go to see a doctor, a cardiologist, they ask you about your family history of heart disease. The population-wide studies done in twins basically predict that half of the risk for coronary artery disease is actually due to the genetics, that the DNA sequences that you inherited from your parents. So now that we know that half of the risk is due to genetics, how do we then study this? I am sure you probably remember these trees from your high school biology classes, where we look at basically a multi-generational family and look at how the disease occurs and what is unique about the DNA sequences of these individuals. So this is a type of study that we call in human genetics a linkage study. And where what we do is we basically follow a multi-generational family and how the disease progresses and how it co-occurs with the presence of a certain mutation in the DNA sequence. And there are thousands of rare diseases that have been found through these linkage studies. And what we find usually are these really rare DNA variants that you see 1 in 100,000 people, 1 in a million people, and they have very large effect sizes. So if you have the mutation, then you have the disease. But atherosclerosis is a lot more complex than a single gene, single disease kind of an approach. It's actually multifactorial. There's a lot of genes that are involved in atherosclerosis. So in the last 15 years or so, we have been doing these association studies where we look at a large number of people who are either who have the disease or who are healthy. So probably people who have had a heart attack and people who don't have a heart attack, and basically look for common DNA variants that affect this disease. So I'm going to explain to you how we do this in this next slide. So this is a totally random region of a genome of 20 different individuals, okay? And as you can see that the 4 nucleotides, ACTG, are mostly invariant. They don't change across these individuals. Each of these lines, each of these rows is an individual. And we typically carry 3 billion of them. I'm showing you a very, very small portion of this. But you can see 2 different regions here, 2 different letter changes between the different individuals. Here is an individual that has a G here and another individual that has a C here in their DNA sequence. These are what we call single nucleotide polymorphisms. So these are really just a change in a single nucleotide across the population, a population of humans. And here in this specific region, there are 2 single nucleotide polymorphisms. We call them SNPs for short. So there are 2 SNPs in this region. And in a given population, we have millions of these across the different people. This is what makes us different, right? Our hair colors and our eye colors and height, etc., are partially at least determined by these single nucleotide polymorphisms. But if I tell you, if I give you another layer of data and tell you that the people at the top, these 10 people, have suffered a heart attack, and these 10 people at the bottom are healthy. They're age matched, but they're healthy. They did not have a heart attack. You can immediately see an association, right? People who have had the heart attack tend to have the A allele of this SNP in this region, and people who are healthy tend to have the C allele in this region, right? There is no association, if you look at this, in this other SNP. So there are basically frequency differences between these 2 groups of people in terms of whether they have the A or the C allele. Now, you can, of course, calculate a statistic associated with this, and then that's exactly what we do. And if you do that, you will find that the A allele is associated with increased risk for having a heart attack. So we call that a risk allele. And then the opposite one, the other allele, then we call it a protective allele. This is a very genetics terminology, basically, that we're going through right now. So imagine doing this. So we did this just for 2 SNPs, right? Imagine doing this for the millions and millions of SNPs across the entire human genome, right? When you do that, what we essentially do is a genome-wide association study. And we show the results in plots like this, which we call the Manhattan plots, because geneticists think this looks like the silhouette of the Manhattan, basically skyline. Each of these dots are those individual SNPs that I just explained to you. And this is across the entire human genome, starting from chromosome 1 all the way to chromosome 22. And this is the strength on the y-axis of the association between the risk for coronary artery disease and these genetic variants. This is a result from the largest genome-wide association study that has been conducted for coronary disease risk. And it involves about 650,000 people. It's a pretty large study. It took many, many years to do this, basically. And what you see are these towers. These are genomic regions that are associated with the risk of the disease. And this line basically shows the statistical significance cutoff that we use to say, yep, this region is associated with the disease. So if you end up calculating all the SNPs in this top portion, we find that there are about 6,000 SNPs that are associated with coronary artery disease risk in the human genome. Now, what is special about these SNPs? When we categorize these SNPs to see if they are leading to changes in the protein sequence, in the amino acid sequence, only a very, very small portion, only 1% of them lead to changes in the codon structure that would lead to a different amino acid in the proteins. So the majority, a great majority, 99% of them are actually in what we call non-coding regions. These are regions of the human genome that just do not encode for proteins, which is obviously the important building block, if you will, of the cells. So what do? What they do is they affect gene expression, okay? So this is a cartoon. Imagine a case like a person having an A allele in this region right in front of the transcription start site of this gene. So this gene will be transcribed into an mRNA, and then it will be translated into a protein. And then imagine another person with the T allele in this same region, but this person leads to higher expression of this gene. There are many more mRNA molecules in this person, the second person, than this first person. So these are basically what we call expression quantitative trait locus. So these are loci, genomic loci, that are associated with the abundance of genes, how much a gene is transcribed. Now, we take advantage of a property in the human genome that we call pleiotropy to use these eQTLs to understand the disease. So I'm going to give you a hypothetical example where we have a genomic locus that is associated with LDL cholesterol. So LDL cholesterol is the bad cholesterol, basically, that your doctor would prescribe you a medication to lower it because there's a direct correlation between LDL cholesterol levels and coronary artery disease risk. So if the exact same locus is also associated with risk for coronary artery disease, we can immediately put this arrow here and say that the mechanism of this locus is by regulating the LDL cholesterol levels, and that in turn affects the risk for coronary artery disease. So this locus we call pleiotropic because it is associated with two different things, right? The LDL cholesterol and coronary artery disease. We can use the exact same approach for basically gene expression. So if a locus is associated with gene expression and also the risk for coronary artery disease, we can create a statistical model to basically ask if the mechanism of this locus is through the regulation of gene expression at this locus, which in turn regulates the risk for coronary artery disease. So now imagine doing this across the entire genome. This is basically the approach that we take, which we call systems genetics. So you have many, many loci. In the case of coronary artery disease, we have 175 loci that are associated with the disease at the top. But then you have all these intermediate molecules that this information is encoded through. So whatever information is in your DNA sequence, it's affecting transcript abundance, it's affecting protein abundance, they're interacting somehow, they're affecting cellular function, and then they're affecting the disease. So if we do this genome-wide in a large population, then we're essentially doing systems genetics to understand how these loci affect the coronary artery disease risk. So when I started the lab about 5-6 years ago, we had a very simple hypothesis. And our hypothesis was that some of these 6,000 or so SNPs, these variants that are associated with coronary artery disease, are affecting this process of stable fibrous cap formation. Remember, I told you about the vulnerable and the stable plaque. Because in the disease process, what happens is these cells that are in a quiescent state get activated, become proliferative, and migrate to this region to essentially form the fibrous cap. And if you can also imagine that fewer cells here means less stable fibrous cap, which would lead to heart attacks, plaque rupture, etc. So we wanted to understand how some of these risk variants are affecting this process, basically, of switching the cells from a quiescent state to a proliferative state. So for this, we collaborated with heart transplant surgeons at UCLA. And what we did is we collected tissues from heart transplant donors. So these are individuals who unfortunately died in typically car accidents, automobile accidents. And their families were frankly very generous enough to donate their organs to be transplanted into people who are suffering from heart failure. And when the surgeons are transplanting these hearts into the recipients, these people are suffering from heart failure, they have to cut pieces of the aorta to really fit it in to the person who is receiving the heart. And normally they would throw away these aortic tissue, basically. They're discarded during the surgeries. So we collected them, and then we grew smooth muscle cells from these tissues. This is, as you can imagine, a really precious tissue material, because obviously you cannot do this in a living human, because you cannot obtain this tissue from living humans. And so the only way to do this is from cadavers and from heart transplant donors. So we were able to do this in about 150 people. And this took almost 10 years to collect this many people, because heart transplant surgeries are not all that common. And many of them, about 2/3 of them, were male, with about 1/3 of them are female. This is basically what the heart transplant donor profile is in the United States. So the first thing we did was to genotype these individuals for 6.3 million SNPs. Remember, I showed you just 2 of them. So across the entire genome, we obtained information from about 6 million SNPs. And then we collected them, this information, and used a reference population to figure out the genetic ancestry of these individuals. If you have done these genetic testings like 23andMe and other things, you've seen these kind of basically plots. And it turned out that about half of our donors had European ancestry, about 40% had Hispanic and Latin American ancestry, 8% had African ancestry, and 4% had East Asian ancestry. So this is a multi-ethnic donor population. Then we took these cells and essentially did this really large-scale, high-throughput experiment where we cultured the cells and we divided them up into different portions, and we measured 3 different phenotypes that are relevant to atherosclerosis. Remember, I told you that the cells migrate from a quiescent state to the lesion, so we measured their migration capacity. We measured their proliferation rate, because how many smooth muscle cells are in the fibrous cap is really important in terms of the stability. And in advanced stages of the disease, there's calcification that occurs, so we measured how much they calcify. So these are the cellular characteristics that we did. We also did molecular characterization. So first, we measured gene expression, and we measured gene expression in 2 distinct states, both in the quiescent state of the cells and the proliferative state of the cells. We also did some gene regulation experiments where we measured the chromatin, the DNA that's open and accessible, and we also measured the amount of protein that these cells secrete to the media, because the secretion of extracellular matrix proteins is really important in the fibrous cap stability. So I'm mostly going to focus on the gene expression part for this talk, because we already published this top part, and I want to share with you unpublished work. So we did pretty deep sequencing of the RNA, and basically we were able to measure about 13,000 protein coding genes, about 3,000 long non-coding RNA, and about 500 other types of RNA. There's many, many different types of RNA, for a total of 18,000 genes. So we were able to measure the expression of 18,000 genes in 151 different individuals. And as you can see here, when we cluster the gene expression, it clusters into these two major categories, the quiescent and proliferative categories, which is exactly what we would like to see, because this tells us that there's different genetic regulation and there's different gene expression regulation going on in these two different conditions. There is a publicly available data set that's called GTEx. This is Genotype Tissue Expression Project. Some of you, those who do bioinformatics, may have already used this, but this is a project where about 45 different tissue samples from about 1,000 different people have been collected, and their gene expression has been measured. And this is a publicly available data set. So we clustered the gene expression of our cells, which are in this corner here, with the 45 different tissues from this publicly available data set, to understand how relevant the gene expression that we're measuring is to in vivo physiology. And to our delight, what we observed was our cells clustered with tissue that is really rich in muscle cells, basically. So, and not blood or not brain and not liver, things like that. So this is telling us that we're at least capturing something relevant to in vivo physiology in humans. Next, we did this expression quantitative locus discovery. So remember, I told you that we're basically looking for associations between genotype and gene expression. And we run a linear regression model for 18,000 genes and 6.3 million SNPs, about 500 billion calculations. And the idea is to find out if there's an effect of the SNP on gene expression, irrespective of the other confounders that we know might be affecting gene expression. And we're looking for cases like this, where there's a difference in gene expression between the two genotypes. So when we do that, we discover that about 1/3 of the genes that we measured have an expression quantitative trait locus. And what's also interesting was there was little overlap between the two different states. And many of the eQTLs that we discovered were unique to the quiescent or the proliferative state of the cells. So we tested this more formally using a difference in effect size approach, using statistical approach. And we found cases where there is indeed very unique effects of the genotype based on the phenotypic state of the cells. So here's one example on the left-hand side. For this gene, NDUFA12, the exact same genotype has an association only in the proliferative state of the cells, where there is no association in the quiescent state of the cells. Or for this other gene in the middle, the genotype, the exact same genotype, has an opposite side effect in the quiescent and the proliferative cells, where the T allele is associated with higher expression in the quiescent conditions and lower expression in the proliferative conditions. Or we observe cases where there's a difference in the magnitude of the effect, where in this case, the same signal nucleotide polymorphism variant has a much larger effect on the quiescent phenotype, whereas the much smaller effect on the proliferative phenotype. This is basically a very classic example of a gene by environment interaction that we observe in humans. And this is really the basis of a lot of the pharmacogenetic studies, where you give a drug to someone and they don't respond because they have a certain genotype, and you give the exact same drug to another person and they respond because they have a different genotype, right? So we're observing this widespread gene by environment interaction in our data set. So what can lead to these differences? So one of the ways gene expression is controlled is by the binding of transcription factors to these upstream regions of of the genes to the DNA. So these transcription factors are proteins that basically bind to the DNA and regulate gene expression. So to find out which transcription factors might be responsible for these differences, we took an approach where we first assayed our donors for regions where they have accessible chromatin. So these are basically regions that DNA is really open, where the transcription factor can come and bind. And these are represented by these peaks. And we use an assay called ATAC-seq to identify these regions. So once we focused on these regions, we also looked at the DNA sequences in these regions and the transcription factor binding sites in these regions, and looked specifically for DNA variants that might be affecting the transcription factor binding site. So we do this for the 6,000 or the 1,000 or so eQTLs that are different between the different conditions, and then we calculate their enrichment for the binding sites. And what's really interesting is, as you can see, that there are 5 different transcription factors that have very different but significant enrichment between the 2 different conditions. For example, SP2 transcription factor binding site seems to have much less enrichment in the quiescent state, but a lot more enrichment in the proliferative state. And this transcription factor has never been shown to play a role in this phenotypic switching of smooth muscle cells from a quiescent to a proliferative state. So we think that these are really interesting master regulators, if you will, of gene expression in these 2 different phenotypic states in smooth muscle cells. Remember, I told you that about 1/3 of our donors are female and about 2/3 are male. So we looked to see if we can identify any sex-biased eQTLs. And to our surprise, we were able to see some really strong effects. So again, you can see that the expression of this gene is associated with the genotype only in females, but not in males. In this specific case, for this gene expression, this AC005 gene, this genotype has a totally opposite effect in females and males. And again, we find this magnitude difference between females and males. And again, in this case, there's a larger effect of the genotype on female gene expression than male gene expression. So this is something that we're actively pursuing to figure out if these are actually effects of hormones, the presence of estrogen and progesterone, or some other non-hormonal mechanism that's leading to this. But what's unique about this is some of these can explain the difference that men and women have in coronary artery disease risk. So of course, you know, I started with telling you about the loci that are associated with coronary artery disease risk. And I also told you that our approach can perhaps identify some of the mechanisms of these loci. So we, of course, ask this question: if the GWAS loci that are associated with coronary artery disease regulate smooth muscle cell gene expression. So we use a statistical approach called Bayesian colocalization to answer this question. So remember the Manhattan plot that I showed you at the beginning. This is now a zoomed in version to that Manhattan plot. Again, each of these dots are the SNPs that we measured. And you can see that there is a signal here for coronary artery disease risk. And there's a signal here for the expression level of this gene, that this gene's expression is regulated by this gene. So to see if there is a shared genetic signal between this gene expression and coronary artery disease risk, we calculate the Bayesian colocalization and also plot them. And when you plot it, if there is colocalization, you should see a case like this where there is a diagonal showing that this SNP is responsible for both the risk for coronary artery disease and the gene expression. And the main reason this is important is because then we can say, well, then this gene is potentially causal in this locus. It's having a role in smooth muscle cells to cause this disease. So when we do this across the entire genome, we have 85 genes whose expression is regulated by the genetic variants in coronary artery disease loci. So this is a bit of a complicated figure, but let me just try to explain this. So what you're seeing here on the x-axis is basically the location of the locus across the entire genome from chromosome 1 to chromosome 22. And this is, on the y-axis, is the effect of the SNP on the gene expression. And the way we did these is basically we took the risk allele, the allele that increases your risk for coronary artery disease, and divided the plot into those that have increased expression with the risk allele, and those genes would have lower or decreased expression with lower risk. So if you are a pharmaceutical company, right, you would want to basically decrease the expression of these genes if you want to prevent coronary artery disease, or you want to increase the expression level of these genes at the bottom to prevent or to treat coronary artery disease. So the eQTL studies allow us to create hypotheses based on directionality with respect to the risk allele. So if we categorize these genes, these 85 genes, based on which condition we find them to be, what is unique is about half of them are common in both conditions, in both the quiescent state of the cells and the proliferative state of the cells. But there are also genes whose expression is regulated only in one condition, let's say in the quiescent condition or the proliferative condition. So this actually tells us the complexity of these loci and how they might be affecting the disease risk. They might start affecting the disease risk really early on when the cells are still in a quiescent/healthy state, or they can start being active in the advanced stages of the disease when the cells have reached to this proliferative state. So before I show you this next slide, so of course, studying these 85 genes one-on-one is an approach that, we are certainly taking, at least for some of them. But it's a lot of work, right, to really try to understand every single one of them, to create mouse models, to do a lot of experiments. Each of them are essentially a PhD project, if you will, to understand how they're affecting the disease risk. So we take a more systems biology approach and try to understand how these 85 genes actually interact with each other to increase the disease risk. And the approach that we take is an approach that we call Bayesian network formation. So in this approach, we have the expression levels of the genes across the entire population, as you can see in this plot. And we can use the conditional probabilities and the Bayesian formulas, basically, to add these arrows and to reverse engineer this network from the state of each node and try to understand the interactions between these genes. And we can even go one step further and say that, for example, this gene, gene C, regulates the expression of gene D, or gene B regulates the expression of gene D, right? So we can create these network maps using the data that we have. So I'm going to show you the network that we created from the 18,000 gene expression that we did. Of course, this is going to be pretty meaningless because you can't read this. It's a huge network. It shows all the interactions among smooth muscle cell genes. And the ones that are in red color are actually the 85 genes whose expression is regulated by the coronary artery disease loci. And the ones in these teal color are the ones that are the upstream regulators of these genes that we think are playing an important role in the coronary artery disease process. So one of them is a gene called Mia3. So this is the last part of the talk, because I want to show you how we then make sense of these basically systems biology predictions. So I showed you these plots before. You can see that the expression level of the gene has the same association with the coronary artery disease risk SNPs. And so this tells us that the risk at this locus is through the expression of this Mia3 gene. You can see that the risk allele, in this case G, leads to lower expression of Mia3 in our smooth muscle cells. Remember, we also measured these phenotypes in these cells, the migration, proliferation, calcification phenotypes. So we can also figure out if there's an effect of this risk allele on a phenotype, smooth muscle cell phenotype that's relevant to atherosclerosis. In this case, it turns out that there's an effect on proliferation. So the risk allele is associated with lower proliferation of smooth muscle cells. And as one would expect, then there is a positive correlation between the gene expression and the proliferation. So we analyzed data, single-cell RNA-seq data from human coronary arteries. These are thousands of cells that have been isolated from 4 human coronary arteries. And there are many different cell types in the human coronary arteries in disease state. And you can measure all these gene expression in this disease state. But the one that I'm showing in this circle are the smooth muscle cells. And you can see that our gene is expressed in the smooth muscle cells, not only in cell culture as we found, but also actual human coronary arteries. This gene was pretty interesting to us because it is actually playing a role in secretion of this extracellular matrix protein called collagen into out from the smooth muscle cells. And these collagens are really important in creating stability of the fibrous cap and preventing a heart attack. So what we did was we lowered the expression of this gene using these shRNAs in cell culture, which led to lower proliferation as we would expect from our human genetics findings. So we can corroborate our predictions basically using in vitro experiments. We also looked at a mouse model of atherosclerosis. So you can see this is a mouse lesion. When we delete this gene called Oct4 in smooth muscle cells, it leads to this thick fibrous cap compared to this thin fibrous cap. And this is basically a stable lesion. This is a vulnerable lesion. So we use this mouse model in collaboration with Gary Owens' lab here at UVA and stained these lesions for our gene of interest called Mia3. These are fluorescence images. I apologize, they're not great, but what we're really looking for is the expression of this gene Mia3 in the fibrous cap lesion in smooth muscle cells. And what we find is that fewer smooth muscle cells in unstable lesions expressed this gene, this protein Mia3, which is again in agreement with our human genetics findings that says that the risk allele is associated with lower expression of this gene. And finally, we actually went to collaborated with the CV Path Institute and took coronary artery samples from people who died from sudden cardiac arrest. And we looked for lesions in their coronary arteries with these thick fibrous caps. So these are the stable lesions, and thin fibrous caps. These are the unstable lesions. And stained for this gene of interest, this Mia3, in the thin and the thick fibrous caps. And what's really remarkable is basically the cells in the thin fibrous cap, fewer cells, were positive for our protein of interest, in agreement with our human genetics findings and mouse findings. So in the interest of time, I'm just going to skip this next section and just summarize that what we found is these coronary artery disease loci regulate the expression of 85 genes. And this specific locus, this Mia3, fell out from our network studies showing that it is a key regulator of smooth muscle cell proliferation. I don't have time to show you this, but it's really interesting that some of the loci actually affect the splicing of the genes with different transcript isoforms. So finally, I'd like to thank the people who have been involved in this project. This is a postdoc in my lab, Dr. Redron Ahararu. He really very painstakingly cultured these cells in hundreds of cultures and did all of these experiments. Here you can see him finally really happy at the end of his experiments and content that these experiments are over, basically. We had a lot of collaborators that shared data with us or helped with different techniques and our funding sources. And I also want to acknowledge several people in the lab who have been involved in various aspects of the project, but especially these undergraduate students, Erin, Diana, Nagazi, Rita, and Dylan, who have done various parts of this project really, really, and carefully. And with that, I would like to thank you again for your attention and listening to me during your dinner time, and happy to answer any questions.
Thank you so much, Mete. I just want to remind our, I think you can type in your questions in the Q&A, and I do see a comment and a question. If you can see them, Mete.
Yeah, I can.
Yeah.
Should I read it briefly and then answer that? Or do you want
Yes, please. Okay. No, you can read it.
Okay. So the question is, have you ever tried, or is it in your agenda from your screening experience using organoids or microfluidic chips? So let me first answer that. Yes, actually, this is an outstanding question. The reason is because obviously we're lacking the cell-to-cell interactions that would occur normally, right, in an artery in our 2D cultures. So we are collaborating with Novo Nordisk, and they have a microfluidic device where they can culture endothelial cells and smooth muscle cells. And we have these endothelial cells. These are the cells that are in the inner lining of the aortas. And basically try to see the cell-to-cell interaction and essentially do this experiments again in these microfluidic chips. But that's another several years of work that's going to happen, basically. So maybe in the next 3-4 years, if I come back again, I'll tell you about those results. Can we learn more about the stability of the transcription factors, how it's influenced by microRNA, and how do you control the microRNA to protect their phenotypic state in your assays where you have high throughput samples? This is, again, a really fantastic question. So the short answer is that we can't fully control the microenvironment, of course. But the way we do these experiments are in batches, and we took one donor, cells from one donor, and we cultured them, and we grew a lot of them, and then we froze them. So in each experiment, we take that donor and do the experiments along with everything else, every other donor, maybe 10 additional donors. And basically that donor is our reference donor. So we normalize everything to that reference donor. And that way, we're hoping to essentially take care of this environmental variation, day-to-day batch variation that's happening in the experiments. And the second one is, of course, we're doing this statistically too. We're adding confounders to our linear regression models and trying to take care of it in a statistical way. Another question is there any other human disease caused by Mia3 gene? That's another great question. Not that we know of, but the, I guess, regulation of this gene has, is dysregulated in certain cancers, but it hasn't been shown that it's causal for cancer, that it causes cancer. So it might be just a byproduct of some of the, the changes that occur due to cells becoming like cancer cells, basically. But we don't know of any others. So great questions. And thank you for your attention. I really appreciate these questions.
Yeah, there is one more, Mete, from our students.
What
can you see it?
Uniform sequence which puts the pathogenic SNPs there? Oh, okay. So I think I was not clear about this part. So so these are not somatic mutations. These are basically the DNA sequences that you inherited from your mom and dad, and they don't change throughout your lifetime, right? So, you know, half of your DNA comes from your mom, half of it comes from your dad. And, you know, because we are all polymorphic, we all have hundreds of thousands of these differences in, you know, in our DNA compared to each other. These basically just happen by chance that you inherit these genetic variants. What you're thinking maybe is somatic mutations that occur in cancer, but these are basically germline variants. But great question.
Yeah, Meta, I have a more general question.
Sure.
So maybe I missed it, but you know, because this is not really my field, and I was maybe lost in the details. But is there any pathway from what you learn about these gene expression profiles that would lead to a therapy at the end?
Ooh, I absolutely love that question. Yes. So the reason I didn't show that is because these are predictions and we haven't made any, you know, experimental validation. But, you know, the networks that I showed you, if you focus on neighborhoods of genes, which basically have enrichment for these coronary artery disease genes, one of the pathways that pop up is nitric oxide metabolism pathway, okay? And so the short answer is that's just a prediction. We don't know what it's doing. Of course, nitric oxide metabolism has been shown to be important in endothelial cells because it affects the contraction and relaxation of the smooth muscle. But it also has these mitochondrial connections. So there's a possibility that it's affecting the mitochondrial function in these cells, leading to reactive oxygen species accumulation and things like that, which you are very familiar with. So it's just a prediction. We don't have validation yet, but that's one pathway that really is very significant.
Mm-hmm. And yeah, thank you.
Oh, sorry, there's another question. tRNA sequences of them causing differentiation. I mean, that's certainly a possibility. I don't think we've found any tRNA genes that are associated with coronary artery disease loci, although there are certainly some genes that are in transcription pathway that affect transcription that are RNA binding proteins, so they may be affecting the mRNA stability, but we haven't found anything specific to tRNAs.
Right.
Thank
So
you.
it seems no further question from audience.
I think this was all and very exciting.
Thank you all.
Yeah, thank you so much.
Thank you for the invitation and you know, attending the talk during, you know, a late night in Istanbul. So I really appreciate it. It was wonderful to meet you and see you all.
Thank you very much.
Same here. Same here.
All right, bye everyone.
Bye.
Bye.